So we are completely conditioned to look at tissue repair well, like a mechanical process. You get a cut, blood clots, a scab forms, and beneath that scab, it's just cellular mortar being laid down until the wall is patched.
Right, the classic construction site analogy, just mindless drones pouring concrete.
Exactly. But when you zoom into the microscopic architecture of our organs, that static mechanical view completely falls apart. It is this hyper-communicative, highly regulated ecosystem where the cells building the scaffold are like constantly whispering to the immune system.
And if those whispers are misinterpreted, or if, say, the wrong cell overhears the conversation, the consequences dictate the difference between a perfectly healed organ, a catastrophic inflammatory cascade, or even a deadly tumor.
Which brings us to our mission today. Welcome to another deep dive. Today, we are tearing apart a genuinely monumental 2026 paper published in Science by Thomas Yip and colleagues. They uncovered this hidden cellular neighborhood right inside the pancreas.
Yeah, and it's a neighborhood where an extraordinary feedback loop basically controls the fate of the entire organ.
For everyone listening, you probably track the basics of immunology and tissue biology. So we are going straight into the deep end of the molecular mechanics here. We've got high-level evidence, in vivo murine models, and huge translational potential to cover.
Absolutely. So the paper focuses on an axis between two specific cell populations. On one side, we have ILC2s.
Right, group 2 innate lymphoid cells.
Exactly. These are specialized tissue-resident immune cells, so they don't patrol the blood. They park themselves in specific organs and just, well, stay there. And on the other side of this axis, we have fibroblasts,
Which you can think of as the primary structural architects of the tissue. But I'm trying to visualize this space. Like if I look at the exocrine pancreas, the vast majority of the organ that synthesizes all those digestive enzymes, where exactly are these ILC2s sitting?
Well, to find them, the research team couldn't just use standard tissue slicing. You'd lose the entire three-dimensional architecture doing that.
You just get a flat, dead cross-section.
Right. So instead, they used a technique called optical clearing combined with two-photon microscopy. Essentially, they use chemical solvents to literally strip away all the lipids, the fats, from the pancreatic tissue.
Because the lipids are what scatter the light and make tissue opaque, right?
Spot on. By washing them out, the pancreas becomes quite literally transparent, like glass.
Oh, wow. So you can shine a laser directly into a fully intact 3D pancreas.
Precisely. And when they mapped the ILC2s in this transparent organ, they didn't find them scattered randomly among the enzyme-producing acinar cells. They were highly concentrated in the pancreatic capsule.
The capsule being the thin outer membrane wrapping the organ.
Yes, and also in the delicate interstitial spaces, the little highways stretching between the lobes, so they established this very specific, restricted anatomical niche.
It brings up the second half of the equation, the fibroblasts. But I mean, treating all fibroblasts as generic structural spackle is kind of an outdated concept now, isn't it?
Oh, it is entirely outdated. So to figure out who the ILC2s were interacting with, the team deployed single-cell RNA sequencing, scRNA-seq.
Right, the barcode method.
Exactly. You take the pancreatic tissue, dissociate it into individual cells, and encapsulate every single cell into its own microscopic oil droplet with a barcode. You burst the cell, capture its mRNA, and sequence it.
Which is amazing because rather than seeing a blended smoothie of the whole pancreas, you get a distinct genetic fingerprint for every single cell.
And that fingerprinting revealed something striking. The fibroblasts living right next to the ILC2s in that capsular niche were completely distinct from the fibroblasts living deep inside the tissue.
Wait, really? How different were they?
Genetically very different. This capsular subset expressed a specific surface protein called Pi16, as well as markers like DP04 and IL-6C. So the researchers termed them Pi16 positive fibroblasts.
Okay, let's anchor this for the listener with a visual memory tool. I want you to picture this capsular space as a nursery. The Pi16 positive fibroblasts living in this nursery aren't your standard mature structural cells laying down heavy collagen. They are progenitors. They act as biological seeds.
That is the crucial distinction. Because they have progenitor capacity, these Pi16 positive cells retain the flexibility to multiply and physically mature into a different cellular state.
So they are like stem cells, kind of?
Sort of, yes. They are functionally distinct from the mature fibroblasts, the ones providing the heavy-duty structural support deep inside the main body of the pancreas. Those mature cells lack those progenitor markers entirely. Instead, they express genes like Col15A1 and ENG, and they are I6C negative.
But hold on. Just because you have a group of seed cells in the outer capital and tree cells deep in the tissue, how do we confidently prove that one turns into the other? I mean, biological proximity doesn't automatically equal lineage.
Fair point. To definitively prove that the seeds grow into the trees, the team engineered a sophisticated lineage tracing model in mice. They used what's called a DPP4 Crete2 mouse model.
Okay, that's a mouthful. Break that down for us.
Basically, they introduced a genetic modification that allowed them to permanently tag only the cells expressing that early marker, the pi16 positive progenitors, with a fluorescent reporter protein. Once they give the mice a specific drug to flip that genetic switch, that specific cell glows red.
Ah, I see. And because it's a permanent genetic edit in the DNA of that specific cell, any daughter cell it produces through division will also inherit that red glowing tag.
Yes, regardless of what it eventually matures into, they basically attached an inescapable molecular GPS tracker.
That is so clever.
It is, and over time, they observed those glowing red progenitor cells lose their original ILI6C marker, migrate deeper into the pancreatic parenchyma, and literally turn into the mature Col15A1 positive structural fibroblasts.
Okay, let me make sure I'm mapping this algorithm out correctly in my head. We have an established nursery in the outer capsule. The ILC2 immune cells are living shoulder to shoulder with these pi16 positive progenitor seeds. But cells don't just spontaneously decide to grow and migrate. There has to be a trigger mechanism.
Right, there's always a signal. The trigger here is an elaborate molecular alarm system reliant on a protein called IL-33.
IL-33, okay.
And what is fascinating about IL-33 is how it's stored. These pi16 positive progenitor fibroblasts produce massive amounts of IL-33, but they don't secrete it into the environment. They lock it away tightly inside their own nucleus, literally bound to their chromatin.
Which makes sense as a safeguard. You don't want a massive inflammatory response just because a cell got slightly stressed. The cell basically has to be ripped open or undergo severe necrotic death for the nucleus to degrade and release the IL-33 into the extracellular space.
Which is exactly why immunologists classify IL-33 as an alarmin. It is a damage-associated molecular pattern. It only sees the light of day when catastrophic structural damage occurs.
So an injury damages the pancreatic capsule, those progenitor fibroblasts die, their nuclei rupture, and a flood of IL-33 is released into the local microenvironment.
Yes, and who is sitting immediately next door equipped with the exact receptors needed to detect that alarmin?
The ILC2s.
Exactly. The ILC2s are highly sensitive to IL-33. The moment that alarmin binds to their surface receptors, the ILC2s activate and begin pouring out their own signaling molecule, a cytokine called IL-13.
So it's a feedback loop. The dying fibroblast sets off the fire alarm. The immune cell hears it and responds by broadcasting IL-13. What does that IL-13 actually do?
It acts directly back on the surviving pi16 positive progenitor fibroblasts in the nursery. IL-13 is a highly potent growth factor for these cells. It forces them into the cell cycle, causes rapid proliferation, and initiates the differentiation program that turns them into the mature Col15A1 positive cells needed to patch the damaged tissue.
To prove how vital this loop is, the paper looked at knockout mice, specifically mice genetically modified to completely lack ILT2s, the IL-7RCR plus Roroflin mice.
Right, and when you remove that immune cell middleman, the nursery essentially goes barren. The numbers of both the progenitor and mature fibroblasts drop severely.
Meaning this IL-33 to IL-13 conversation isn't just for emergencies. It's like a constant baseline hum required just to maintain normal organ structure.
Exactly. Without the ILC2s constantly interpreting minor, everyday cellular wear and tear and sending that low-level IL-13 growth signal back, the pancreas just cannot sustain its own architectural scaffold.
Okay, this is a lot of dense biology, so let's do a quick recap for the listener before we move on. We promised an algorithm, so here's the step-by-step. Number 1, in the nursery capsule of your pancreas, you have progenitor pi16 positive fibroblasts. Number 2, when they die, they release a nuclear alarm, IL-33. Number 3, that wakes up ILC2 immune cells. And number 4, the ILC2s send a chemical text message, IL-13, back to the surviving fibroblasts, telling them to multiply and mature to rebuild the organ.
That is a perfect summary.
But that raises a massive question. I want you to think about this. If IL-13 is such a potent signal for proliferation, there has to be a biological kill switch. If the ILC2s are constantly telling fibroblasts to multiply, why doesn't the pancreas eventually turn into a dense fibrotic rock? What stops this loop?
That is the multi-million-dollar question in fibrosis research. Identifying that brake pedal required an entirely different, incredibly elegant technology known as lipstick.
Lipstick. I love these acronyms. What does it stand for?
Labeling immune partnerships by sortagging intercellular contacts.
Say that 5 times fast. So how does it work?
They modified the mice so that whenever an ILC2 physically bumps into and touches another cell's membrane, it leaves behind a permanent biotin molecule. It's basically a molecular smudge that says an ILC2 is here.
Oh, that's brilliant. So they can map exactly which cells the ILC2s are physically restraining through direct contact.
Well, that was the hypothesis, but when they analyzed the tissue, the fibroblasts were completely clean. The ILC2s were not physically touching the fibroblasts to shut them down.
Wait, really? Then who was?
Another cell population entirely was heavily tagged with those molecular smudges. A specific subset of tissue-resident macrophages marked by the protein TIM4.
Okay, TIM4 positive macrophages. Now, we normally think of macrophages strictly as the immune system's cleanup crew, right? Engulfing foreign pathogens or clearing out dead cell debris. But here they are performing routine architectural maintenance.
They act as the ultimate population control. Think of them like Pac-Man. The TIM4 positive macrophages physically interact with the Pi16 positive progenitor fibroblasts and regulate their numbers through direct phagocytosis.
So they are literally eating them. They consume the excess progenitor cells to ensure the nursery doesn't overpopulate.
Exactly.
But wait. If the macrophages are eating the fibroblasts, doesn't that rupture the cell, break open the nucleus, and release more of that IL-33 alarmin? Wouldn't that just create an infinite loop of inflammation?
And that is exactly why the mechanism of cell death matters so much here. Phagocytosis by a macrophage is an immunologically silent process. The macrophage fully engulfs the intact fibroblast and digests it internally using its lysosomes. The fibroblast's membrane never bursts into the extracellular space, so the IL-33 alarmin is degraded safely inside the macrophage. No alarm is ever sounded.
Wow, that is such an elegant design. It trims the population without triggering the fire alarm. And the paper also notes that while macrophages handle the progenitor seeds, the ILC2s themselves exert a distinct indirect braking force on the fully mature Col15A1 positive cells.
Yes, likely using IL-6 family cytokines, possibly oncostatin M. So the ILC2s operate as the master control dial. They turn the system up by releasing IL-13 to drive proliferation when damage occurs, but they coordinate with macrophages and release secondary signals to cap the population of mature cells, preventing fatal fibrosis.
It is a perfectly calibrated system until, of course, you introduce a catastrophic injury that completely overwhelms the balance.
Right, and that leads us to the disease models.
Let's explore that, because we want to make this clinically relevant. Let's introduce a hypothetical clinical case. Let's say a patient, John, comes into the ER with severe abdominal pain radiating to his back. He's diagnosed with severe acute pancreatitis. The researchers wanted to see how this ILC2 fibroblast system reacts to a massive acute organ injury like that.
To model that in mice, they use a well-established chemical trigger called caroline.
Caroline, what does that do?
Caroline overstimulates the acinar cells in the pancreas, causing them to prematurely activate their digestive enzymes while they are still inside the tissue. So instead of flowing into the gut to digest food, these highly corrosive enzymes begin digesting the lipid bilayers of the pancreas itself.
Literally a chemical burn from the inside out.
Yes. It causes profound, sweeping tissue necrosis within 24 hours.
In that environment, millions of those Pi16 positive fibroblasts are going to be shredded simultaneously.
Which means a tidal wave of the IL-33 alarmin is dumped into the interstitial space all at once. The ILC2s are hyperactivated, screaming back with IL-13, demanding massive tissue regeneration.
And here is where the paper delivers a massive conceptual twist. To isolate the variables, the researchers induced this severe pancreatitis in the genetically modified mice that lack ILC2s. As we established earlier, without ILC2s, these mice have a barren nursery. They have far fewer fibroblasts at baseline.
Right, and logically, you would assume that a pancreas with less structural scaffolding would be more fragile and the injury would be much more devastating.
But the data showed the exact opposite. The mice with fewer baseline fibroblasts actually experienced significantly less acinar cell necrosis during the initial early stages of the pancreatitis attack.
It completely upends how we view these structural cells. The researchers realized that the baseline density of your fibroblasts establishes your inflammatory set point, or what they call the tissue tone.
Because every single one of those progenitor fibroblasts is basically a microscopic powder keg loaded with IL-33.
Yes. The more fibroblasts you have, the more explosive the environment. When the initial Carey Lane injury hits, the sheer volume of IL-33 and other DAMPs like HMGB1 released by the dying fibroblasts triggers a massive, disproportionate overreaction from the broader immune system.
So it's collateral damage. The immune system rushes in so aggressively to respond to the alarm that the subsequent sterile inflammation actually destroys more surrounding tissue than the initial chemical burn did.
It is a profound double-edged sword. You absolutely require that dense population of Pi16 positive progenitor cells to eventually rebuild the tissue in the days following the injury. But possessing that robust rebuilding machinery is exactly what fuels the catastrophic inflammatory fire during the initial insult.
This concept of tissue tone fundamentally changes how we approach acute organ injuries clinically. The severity of the attack isn't just about the external trigger. It is dictated by the baseline architecture of the organ before the injury even happens.
It does. Which logically forces us to ask an even darker question. What happens when this powerful, explosive rebuilding machinery is manipulated by a permanent, highly localized threat?
Okay, let's do our second mini recap before we dive into the dark side of this.
Yeah.
Bullet 1, acute injury happens. Bullet 2, fibroblasts die and release massive alarms. Bullet 3, more baseline fibroblasts equal a louder alarm, which equals more collateral tissue death early on. But bullet 4, you need that system to eventually trigger the Pi16 positive cells to rebuild.
Perfect.
So my question to you, the listener, is this. Knowing this, what happens if this powerful, explosive rebuilding system is tricked by a permanent injury? Like a genetic mutation that won't go away.
You're talking about a scenario where the alarm is jammed in the on position.
Exactly. And that leads us directly to the most clinically urgent section of this research. Pancreatic ductal adenocarcinoma, or PDAC. Pancreatic cancer is historically one of the most lethal malignancies, largely because of how it physically alters the organ.
PDAD is notorious for its stroma. A pancreatic tumor doesn't just grow as a ball of mutated cells. It surrounds itself with an incredibly dense, rigid matrix of fibrotic scar tissue.
It's essentially a physical fortress.
Exactly. It collapses the local blood vessels, which prevents chemotherapy from reaching the tumor, and it physically excludes cytotoxic T cells from attacking the cancer.
But the tumor isn't building that fortress out of nothing. It requires massive amounts of mature fibroblasts to lay down that dense collagen matrix. And based on everything we've just unpacked, we know exactly where the pancreas keeps its supply of pre-programmed architectural seeds.
Right. So the researchers utilized advanced genetically engineered mouse models of pancreatic cancer, the KPC and KC models, which mimic the slow, stepwise progression of human pancreatic cancer. They tracked the Pi16 positive progenitor niche from the very earliest stages of precancerous lesions, which are known as pennions.
And as those precancerous cells begin to grow and damage the local architecture, they are essentially pulling the fire alarm.
The local microenvironment becomes flooded with inflammatory signals, including our alarmin, IL-33. The ILC2s detect the alarm, assume there is a massive wound that needs immediate repair, and begin pumping out IL-13 to activate the nursery.
But a tumor isn't a transient wound that eventually heals. It is a chronic, expanding source of damage. The alarm never shuts off.
And this is where the lineage tracing tool revealed something truly terrifying. Under the relentless pressure of this tumor-driven inflammatory loop, those glowing red Pi16 positive progenitor cells did not mature into normal, healthy structural trees. The cancer microenvironment forced them to transdifferentiate.
They mutated.
They turned into cancer-associated fibroblasts, or CCAFs.
The cancer is literally conscripting the body's own repair crew. It sounds the alarm, tricks the ILC2s into rapidly expanding the progenitor population, and then fundamentally rewrites the programming of those new cells to serve the tumor.
Yes. And single-cell RNA sequencing confirmed that these hijacked progenitors differentiate into highly specialized malignant subtypes.
What kind of subtypes?
They become ICTEFs, or inflammatory CTEFs, which secrete cytokines that further suppress the immune system. They become MySAFs, myofibroblastic CHAFs, which are highly contractile cells that physically squeeze the tissue, generating the intense solid stress that collapses those blood vessels.
They also identified a novel subset in the paper, the SOC6 positive CFs, which proves that this hijacked nursery generates an entire specialized workforce dedicated solely to protecting the malignant cells.
But the most powerful finding in the paper came when they tested this progression in mice lacking the ILC2 immune cells.
Going back to the knockouts.
Right. Without the ILC2s present to interpret the IL-33 alarm and broadcast the IL-13 growth signal, the density of those cancer-associated fibroblasts plummeted significantly.
So by removing the cellular middleman, They effectively cut the communication line. The tumor was still trying to sound the alarm, but there was no one to answer it, and as a result, it couldn't conscript the fibroblasts to build its fortress.
It proves that the very same physiological axis, the ILC2 and fibroblast conversation, that maintains the delicate architecture of a healthy organ, is the exact mechanism hijacked to construct the lethal stroma of pancreatic cancer.
This research really is a masterclass in biological duality. It forces us to recognize that evolution doesn't invent entirely new pathways for disease. Pathology is almost always born from the dysregulation of our most essential, elegant healing mechanisms. The communication loop that keeps your pancreas intact is the exact same loop that dictates the collateral damage of acute pancreatitis and the exact same loop that builds the armor for a deadly tumor.
And from a therapeutic standpoint, this provides an incredibly precise target. If pharmacologists can design small molecules or monoclonal antibodies that interrupt this specific node,
like blocking the IL-33 receptor on ILC2s or neutralizing the IL-13 signal itself.
Exactly. If we can do that, we could potentially dictate the fate of the tissue.
We could theoretically dial down the explosive tissue tone in a patient presenting with acute pancreatitis, preventing the devastating secondary necrosis. Or, perhaps more profoundly, we could administer a drug that silences this communication loop in a pancreatic cancer patient.
Starving the tumor of its architectural workforce.
Right. Stripping away its dense fibrotic armor and finally allowing traditional immunotherapies and chemotherapies to actually penetrate the malignant core.
The clinical potential of dismantling the tumor microenvironment by targeting the host's own hijacked repair mechanisms is really one of the most promising frontiers in oncology today.
It is a stunning mechanical revelation. But as always, we want to leave you, the listener, with a lingering, provocative question to mull over. If we successfully develop a therapy that systematically blocks this IL-33 to IL-13 repair pathway to dissolve a tumor's fortress, we are fundamentally turning off the body's primary mechanism for structural healing.
A systemic block would affect every organ relying on this pathway.
Exactly. So how do we deploy a weapon that starves a cancer of its repair crew without simultaneously stripping the rest of our vital organs of their ability to heal from the everyday microscopic damage of simply being alive?
Right.
Are autoimmune diseases and cancer just two sides of the exact same evolutionary coin?
That tension between eradicating the pathology and preserving the baseline physiology, that will be the ultimate hurdle for the next generation of therapeutics.
It is a delicate, razor-thin line, and it's going to require an immense amount of precision medicine to solve. Thank you for joining us on this deep dive into the hidden microscopic architecture of the pancreas. We'll catch you next time.